So today, just like Dr. Peter said, we're continuing on our sessions on introduction to epidemiology and statistics. And the topic for today is causality, principles and practice. So that's what we're going to be dwelling on in the next couple of minutes. Okay. So by the end of this presentation, I'll expect us to be able to define key concepts of association and causation in epidemiology, to differentiate between association and causation, to explain the historical evolution of causal networks, including Henley Cox postulates and Sir Austin Bradford Hill's viewpoints, and then to apply the Hill's criteria for causation to evaluate epidemiological evidence, and then finally understand modern causal models and their practical implications for public health action. Okay. So a fundamental challenge. Let's see what is the difference between association and causation. When do we say a relationship is an association, and when do we refer to that relationship as being a causal one? First of all, why is this important? It's important because when we collect data, when we observe data, we definitely see a relationship between variables that have been collected. But this relationship that we see does not automatically translate to saying that one thing causes the other. So a relationship can be seen, so acting on mere association can lead to wasted resources and ineffective and harmful public health policies. So key definitions here to include will be: association is a statistical relationship between two variables. So I'll give us an example. When you go to a very hot climate, you might see that the sales of ice creams, it's very prominent in that place. And that might coincide to the period where flowers are blossoming in that same zone. So probably a researcher that is collecting statistics on flowers and the sale of ice creams might see a relationship between blossoming of flowers and sales of ice cream, and we'll say you can infer that there is an association between blossoming of flowers and sales of ice cream. But that doesn't tell us exactly that the blossoming of flowers is causing an increase in the sales of ice cream. So that's an association. But when we refer to causation, we're actually saying that a change in one parameter or one variable results in a change in another variable. So for example, when you see clouds gather in the sky, and then it subsequently rains, we can say the clouds that gathered produce the rain. So that looks more like a causal relationship than the association relationship of the flowers and ice creams. Okay. So let's look at components of causation. Let's have a very good definition of cause. So according to a book called Essential Epidemiology by Webb and Bain, it defined a cause as an event, condition, or characteristic, or a combination of these factors that play an essential role in producing an occurrence of disease. So we're not limiting a cause to an organism. For example, when you talk about infectious diseases, but it can be an event, maybe for example, if you have an accident and an accident leads to you having an injury. So it can be an event, it can be a condition, it can be a genetic condition that has predisposed one to developing a certain disease, or can be characteristics which play an essential role in producing an occurrence of disease. So causes can be categorized into three major types. So we have what we call a component cause. So a component cause is a factor that contributes towards disease causation, but is not sufficient by itself to cause that disease on its own. So I'll give us an example of a component cause. So when you have tuberculosis as a disease, there are several things that can lead to tuberculosis. First, you have the infectious agent, and then you have that individual needs to be susceptible to that disease. So maybe the individual is malnourished, or is immunocompromised, or is living in a very poor socioeconomic environment, and all of that. So each of these things I've mentioned are component causes. So they all contribute towards the causation of the disease, but each one is not by itself sufficient to cause that disease. So that's what a component cause is. Then we have sufficient cause. Sufficient cause is defined as a factor, or more often, a combination of several factors that an assemblage of them will inevitably produce a disease. So just like I've explained with the TB scenario, before you can have tuberculosis occur in a person, all of these component causes will have to be present in order to be sufficient to produce that disease. The absence of all of these factors might not be sufficient enough to cause that disease. So that's what a sufficient cause is. And then finally, we have necessary cause. So a necessary cause is any agent or component cause that is required for the development of a given disease, in which its absence cannot produce that disease. So the absence of a necessary cause will not produce that disease. So a typical example is if an infectious disease, if the organism that causes that disease is absent, then that disease cannot happen. So let's give malaria, for example. If you do not have Plasmodium species as one of the component causes, or as one of the sufficient causes, then the person cannot have malaria. So Plasmodium is a necessary cause in the causation of malaria. So those are the three component causes. So if you look at the diagram on the right, there is three different pie charts. So if you look, the three different pie charts have the letter A in them. So we can conveniently say that A is a necessary cause, because A has to be present in all of this in order for the disease to happen. And then each of these pies are sufficient causes. So each combination of 5 letters need to be present for the disease to happen. So that's a sufficient cause. Okay, so let's move ahead. So causation has been something that epidemiologists have tried to define all along time. So the first attempt that is recorded to kind of characterize causation was done by Henry Koch's postulate, which was happened in the 19th century. So this postulate has 4 components in it. The first is that the microorganism causing this disease must be found in all organisms that have the disease. So if we use malaria, for example, the microorganism that causes malaria, which is Plasmodium falciparum, must be found in every individual that has malaria. So that's what the postulate says. The second one is that that microorganism must be isolated from a diseased host and grown in a pure culture. So again, using malaria as an example, if someone has malaria, then we must be able to isolate Plasmodium falciparum from his blood and be able to grow that on a pure culture. The third component is that this cultured microorganism should cause disease when reintroduced into a healthy host. So with malaria as an example, when we isolate Plasmodium from a diseased person and we reintroduce that Plasmodium into a healthy person, this postulation says it needs to be able to cause the disease in this healthy person. The fourth component is that the microorganism must be re-isolated from the experimentally infected host. So this new person that has been infected and has developed the disease, we must be able to isolate the organism within that person. In as much as this postulates try to define some level of causation or to explain causation, it was limited by some factors. Number one was that it was limited in that we could only apply it to infectious diseases. So when we have non-communicable diseases, it was difficult to apply this postulate. So for example, things like heart disease and cancers that are not caused by infection, it was hard to apply this postulate. And again, it does not account for asymptomatic carriers or host susceptibility. So these two limitations made it difficult to be able to apply this postulate to other diseases that are not infectious diseases. Okay, so going down the line, Mr. Austin Bradford Hill came up with a framework in 1965 for assessing evidence of causality. Now, because the first postulate was lacking in considering non-communicable disease, Bradford Hill focused his framework more on non-communicable and chronic diseases. However, he was quick to mention that his framework were not a hard and fast rule. However, they were just aspects of association to consider when making a causal inference. So Bradford Hill came up with 9 parameters, which we call the Bradford Hill criteria, to be able to assess causality in a given disease condition. And they include the strength of association, consistency, specificity of the association, temporality, biological gradient, or dose response, and then plausibility, coherence, experimental evidence, and analogy. So let's look at each of these components individually. So the strength of association, according to Hill's criteria, was that for you to consider causality, then there must be a strong association between the exposure and the disease. When we were talking about association, we're talking about measures of association like the relative risk, which is calculated from longitudinal studies, like the cohort study, or odds ratios, which are calculated from case control studies. So if an exposure has a strong association with an outcome or a disease condition, then the level of causality is strong. So for example, there's a strong link between smoking and lung cancer with a relative risk of as much as 20 times. Okay, so that's one of the criteria that Hill considered. The second criteria that Hill considered is temporality. And temporality was considered as an absolute prerequisite for causality. Temporality just means that the exposure came before the outcome. So for something to be able to, for you to say something caused another thing, then that exposure must come before the outcome. The exposure must come before the disease. And that is what temporality is all about. So for example, exposure to a virus must come before that illness happens. Using COVID as an example, you must get the the SARS-CoV-2 virus before you can develop the COVID-19 disease. Okay. The third criteria that Hill came up with was consistency. And consistency here means that in different conditions or in different persons, you must be able to identify that same pattern. So for example, if you have something like using COVID-19 again as an example, whether it is in Africa or in Asia or in the Americas or in Europe, once you introduce that organism, there is the likelihood that that organism will result to that disease, irrespective of the location, irrespective of the epidemiological design of the study, irrespective of whatever. Consistency must be maintained. So when there is consistency in terms of exposure leading to a disease, then there is a strong causal possibility in that relationship. Okay, the fourth criteria that Hill considered was specificity. And that means a single cause leads to a single effect. So again, using COVID as an example, if you have the SARS-CoV-2 virus, then it leads to the COVID-19 disease. That's the specificity that, or if you have Plasmodium falciparum, it leads to malaria. It has to be specific in its cause. Okay. Now, the fifth criteria that Hill considered was what we call biological gradient or the dose response. And that means increasing exposure leads to an increasing risk of outcome. So I'll give you a typical example with HIV AIDS. So when you have the virus in your system, the amount of the virus determines the risk of that disease, of you coming down with AIDS, and also determines the risk of you transmitting the disease. And that is why in using HIV antiretroviral therapy as a preventive form of transmission, the drugs decrease the volume of the virus in an infected person, and therefore it decreases the chance of that person transmitting the virus. So that is what this dose response or biological gradient really talks about. So you can have two people get infected with an organism, but the person that gets infected with more dose of that organism is more likely to get that disease. So that's the biological gradient. Then there is also the plausibility, in which case says the association is coherent with existing biological knowledge. So that exposure outcome relationship needs to concur with what is already known in literature, or what is already existing as biological knowledge. Then the other one is coherence, and that means the causal interpretation should not conflict with generally known facts. So we cannot generally know that now Plasmodium falciparum causes malaria, and then you will bring up another conflicting relationship to that. So that causal interpretation should not conflict with the general known fact of the natural history and biology of disease. For example, the decline in lung cancer rates following reduced smoking prevalence adds coherence to the causal argument. Okay, so the next criteria that Hill considered is experiment. Now says evidence from a controlled experiment strengthens a causal inference. Again, if you take, for example, HIV virus in a controlled setting and infect it into a host, then that host is most likely to develop the disease, and that is experiment. The last criteria that Hill considered was analogy, in which for analogous exposures and outcomes, a causal relationship may be considered. Example, if one virus is known to cause birth defect, for example, rubella, it is reasonable to suspect another similar virus might also cause birth defects. For example, another virus in the species of or in the family of rubella. Okay, so applying the criteria. So let's use a case study of smoking and lung cancer and apply this criteria that Hill developed and see how we can use it to establish causality. So for example, the criteria of strength, we have that there's a very strong association between heavy smokers and lung cancer, as much as a relative risk of 20. So that is telling us that there can be some level of causality within that relationship. Consistency. A consistency is observed in dozens of studies across different countries and design that there is a strong relationship between smoking and lung cancer. The third component is temporality. So again, smoking initiation usually precedes cancer diagnosis. So the person would have been smoking for some time before the diagnosis of lung cancer, showing that the exposure came before the outcome. Biological gradient. Now, it's very clear that the clear dose response exists between smoking and lung cancer. So if someone smokes more, so there's this parameter used to define amount of smoking called, is it long pack? I can't remember exactly, something pack years, where someone that smokes 20 sticks a day in 1 year or 10 sticks a day in 2 years. So the number of cigarette sticks one smokes per day and the duration of period that the person smoked that is strongly related to the development of lung cancer. Plausibility. So tobacco smoke contains dozens of known carcinogens that have been linked to causing lung cancer. Coherence. So this explains the rise in lung cancer rate that is parallel to smoking trends. So it's been seen from observation and from literature that the higher or the more people smoke in a community, the more the incidence or prevalence of lung cancer in that community. Experiments, so randomized controlled trials, even though they are unethical, but cessation studies show a reduced risk. So people that have stopped smoking, we've seen that there's reduced risk of developing lung cancer in people that have disease from cigarette smoking. Okay, analogy. Other inhaled carcinogens can also cause lung cancer. So this is analogous to smoking causing lung cancer. So we can see that in conclusion, the totality of the evidence that is available to us using all of these criteria strongly supports a causal relationship between smoking and lung cancer. So that is how we're able to use the Bradford-Hill criteria to kind of establish some level of causality. But again, remember, Bradford-Hill himself said this is not a hard and fast rule. These are just parameters that we use to increase the plausibility of causality between exposure and outcome. Okay, so moving beyond Hill's criteria, some modern causal methods like the counterfactual framework have also been developed. And this is based on 4 parameters. First of all is the fundamental question. And we'll ask what would have happened to the same group of people if they had not been exposed. So if we're able to answer this question, that can begin to lead us to know if that exposure can be said to have a causal effect to the outcome. The next is counterfactual, the unobservable alternate outcome. So what would be the health of smokers if they had never smoked? Would they have developed lung cancer? So that's another question that can help us establish causality. Then there is a factual. This was the observed outcome of people who smoke. And then finally, there's the causal effect. What's the difference between the factual and the counterfactual outcomes? So when these 4 parameters are considered, it could help us to delineate if an exposure is actually is actually causing the outcome. Okay, so there are challenges in establishing causality. And one of the chief ones is confounding. So remember when we looked at sufficient cause, component cause at the beginning, we saw that for you to say something is a sufficient cause, it's a combination of component causes that whose presence make it inevitable for one to develop a disease. So there are times when there could be confounding, a mixing of effects where an apparent association is actually due to a third common factor. The most common example of confounding is seen when a study was done between alcohol, those that drink alcohol, and lung cancer, and where a strong association was found between alcohol ingestion and lung cancer. However, it was later discovered that that's actually a confounding, that the actual thing is that people who drink are also most likely to smoke. So the smoking there is the confounding factor. So confounding is a major challenge in establishing causality. Then the second is bias. So systematic errors in selection of individuals in a study, measurements of and data collection procedures and information can actually distort associations within between exposure and outcome. So those are things to look out for when you're considering causality. And then chance, again, you could see associations that could be due to chance. So usually when we do statistics, we base our inferences on a 95% confidence interval. So that remaining 5% are associations that can happen by chance. So we must also not forget that there is an issue of random error when we are considering causality. Then there is reverse causality, which is incorrectly inferring the direction of a relationship. And finally, we have complexity, where most diseases have multiple components. This is especially seen in chronic disease like cancer, where there is a combination of both genetic and environmental and behavioral factors that all lead to causing that disease. So all of these are challenges that we have to consider when we're trying to establish causality between an exposure and an outcome. So how do we move from evidence to public health action? So establishing causality is the cornerstone of effective public health intervention and could be applied in terms of prevention. So identifying causal factors can allow primary prevention. So for example, tobacco control can help us prevent lung cancer. Sanitation in terms of clean environment can help us prevent malaria. And then vaccination can help us prevent many diseases like we know, measles, diphtheria, and etc. So the next is policy. So causal evidence can inform legislations and regulations. The most common one we've seen is the use of seat belts. So laws institutionalizing the use of seat belts have been used to help prevent accidents, and then air quality standards are also another way to prevent diseases that are respiratory in origin. A third way we can do this is resource allocation. So when we're able to establish causality between exposure and outcome, then it can guide where to invest for maximum health impact. So summaries and key takeaways from our presentation are that association is not causation. That is very, very key. It's a foundational principle in epidemiological reasoning, that because there is an association between two or more things, it does not mean that one thing is causing the other. We must be able to apply epidemiological principles to be able to establish whether there is causality in that relationship. Again, Hill's viewpoint provides a practical, though not definite, framework for judging evidence. So we can use the criteria to establish causality, but it is not a hard and fast rule. It's not cast in stone. So we need to also be able to use common sense and our epidemiological reasoning in assessing these things. The third is that temporality is essential. So we must be able to establish that the exposure comes before the outcome in order to establish causality. Then modern frameworks like the counterfactuals have been able to provide a more formal structure for causal thinking. The ultimate goal of establishing causality is to inform and justify public health actions that will help us prevent disease and promote health. So these are some of the references for this presentation, and thank you very much for listening.